I'm late to tell you about the process of transcription. Well, now that I'm awake, now I can talk to you about the process of transcription. After transcription, the RNA molecule is processed in a number of ways. Introns are non-coding sections of RNA transcript, or DNA encoding it, that are joined before the RNA molecule is translated into a protein that forms a mature mRNA molecule consisting of a single protein coding sequence. Exons are any part of a gene that will encode a part of a final mature RNA produced by the gene after introns have been removed by RNA joining. RNA synthesis involves the normal base pair rules, but the base thymine is replaced by the base uracil. This is the promoter, this is the DNA helix, this is the DNA polymerase, and this helps form this. This is a helicase, and this is like a zipper, and this is initiation. The DNA molecule unwinds, helped by the helicase, and separates to form a small open complex. This is what the circle is. RNA polymerase connects to the promoter and the template strand. This is the terminator, and DNA polymerase. Now we're going to do an example. So the A's turn to U's, and the T's turn to A's, and G's always C, C to G, U. So the A's turn to U's instead of T's now. And then this just goes all the way. So the T's get canceled out. And that is transcription.
Termination. In prokaryotics, there are two ways in which transcription is terminated. In RHO-dependent termination, a protein factor called RHO is responsible for disrupting the complex involving the template strand. RNA polymerase and RNA molecule in RHO-dependent termination. A loop forms at the end of the RNA molecule, causing it to detach itself.
Termination in eukaryotics is more complicated, involving the additional adenine nucleotides at the 3 of the RNA transcript, a process referring to as to explain what the promoter site does. In genetics, a promoter is a region of DNA that starts transcription of a particular gene. Promoters are located near the transcription start sites of genes on the same strand and upstream on the DNA. RNA polymerase make mRNA to DNA. RNA polymerase unwinds the DNA like a zipper. The promoter site splits the strand of DNA open to create the RNA. RNA polymerase eventually reach a stop location, termination site.
The goal of transcription is to make an RNA copy of a gene's DNA sequence. Not all genes are transcribed all the time. Transcription is controlled individually by each gene that carries information used for protein synthesis. Only one strand of the DNA is transcribed.
Now we're going to talk about translation. Translation takes place in the nucleus where the DNA in the cell is located. Building a polypeptides read in 3 groups, and those groups are called codons. Collection of codon amino acid relationships are called a genetic code. It lets cells decode an mRNA into a chain of amino acids. Your cells are making new proteins all the time, and each of those proteins must contain the right set of amino acids, hooked together in just the right order. That may sound like a challenging task, but luckily your cells, along with those of other animals, plants, and bacteria, are able to do it. Transcription, yeah! Transcription, yeah! What is transcription? I'll tell you. Segments of DNA serve as templates to produce what? RNA.
I feel sick.
That's because I'm here. Bacteria.
Good thing I have this antibiotic.
No! Do not! That will kill me! No! Translation. 4 RNA nucleotides are arranged 20 different ways to make 20 different amino acids. Nucleotides base exist in triplets. Triplets of bases are the smallest units that can code for an AA. 3 bases equal 1 codon equal 1 AA. There are 64 possible codes. 61 of them are amino acids, and then 3 of them are stops and 1 is a start.
Translation initiation. Initiation brings together mRNA, tRNA with the first AA, which is also known as an amino acid, and ribosomal subunits. Secondly, small ribosomal subunit binds to mRNA and the initiator tRNA. The start codon is always going to be AUG or UAC. Small ribosomal subunits attached to 5 and mRNA. Initiation continued. The downstream from the 5' end is the start codon AUG for mRNA. The anticodon UAC carries the amino acid methionine. After the union of mRNA, tRNA in the small subunit, the large ribosomal subunit attaches, and then initiation is complete. Translation elongation is when amino acids are added one by one to the first amino acid, which is usually AUG. Remember, the goal is to make a polypeptide. Step 1, codon recognition. mRNA codon in the A site forms a hydrogen bond with the tRNA anticodon.
Step 2, peptide bond formation. The ribosomes catalyzes the formation of the peptide bonds between the amino acid, the one already in place, and the one that's beginning to add. The polypeptides extending from the P site moves to the A site to attach to the new
Translation
AA.
termination. This happens at the stop codon, and there are 3 of them. The stop codons are UAA, UAG, and UGA. These do not code for amino acids. The polypeptide is freed from the ribosome, and the rest of the translation assembly comes apart. RNA polymerase transcribes introns and exons. This is called pre-mRNA. Pre-mRNA never leaves the cell's nucleus. The introns are excised and exons are joined together to form mRNA.
Ribosomes. The 3 tRNA binding sites are: number 1, A site holds tRNA, is carrying the next amino acid to be added. 2, P site holds tRNA that is carrying the growing polypeptide chain. 3, E site, where discharged tRNAs leave the ribosome.
RNA splicing in eukaryotics. In eukaryotics, RNA transcripts have long, non-coding stretches of nucleotides. These regions will not be translated. The non-coding sections are dispersed between the coding sections. Introns, non-coding sections of nucleic acid found between coding regions. Exons, coding regions of nucleic acids eventually. These are expressed as amino acids. There are 3 types of RNA. RNA is a nucleic acid that consists of a long chain of nucleotides. The first type is messenger RNA, also known as mRNA. This RNA molecule sends signals and carries copies of these instructions to the proteins. Ribosomal RNA, also known as rRNA, it's a type of RNA that combines the proteins to the ribosomes. And the last kind of RNA is transfer RNA, which is also known as tRNA. This RNA carries each amino acid to a ribosome during protein synthesis. In translation, there is a codon that contains 3 nitrogen bases that result in a polypeptide. The steps to translation are: each tRNA is folded into a compact shape. An amino acid is added to the end of the tRNA called an anticodon. An anticodon is a sequence of 3 nucleotides located on one end of the tRNA. A polypeptide bond forms between the two amino acids, and the tRNA leaves amino acid behind. And then the anticodon is a 3 nucleotide sequence that is complementary to the mRNA, and it has to start with the codon AUG, which is also known as methionine.
Translation? Translation? What is that? Let me tell you. Translation, a step in protein synthesis when the genetic code carried by mRNA is decoded to produce a specific sequence. Transcription, process of going from DNA to RNA, takes place in the nucleus. Translation is the process of going from RNA to a protein, takes place in ribosomes.
Now we will talk about protein synthesis. A prokaryotic gene regulates and differs from eukaryotic regulations, but since prokaryotics are much easier to work with, we will focus on prokaryotics at this point. Promoters are sequences of DNA that are the start signals for the transcription of mRNA. Terminators are the stop signals. mRNA molecules are long. Ribosomes are the organelle that are inside in all cells, where the proteins are produced. Ribosomes consist of a small and large subunit. The length of RNA differs in each. The larger subunit has 2 binding sites for tRNA. There are 4 steps in protein synthesis. I will be explaining the first one. The first step in protein synthesis is the transcription of mRNA from a DNA gene in the nucleus. At some other prior time, the various other types of RNA have been synthesized using an appropriate DNA. The RNAs migrate from the nucleus into the cytoplasm.
Step 2. In the cytoplasm, protein synthesis is usually initiated by AUG codon on mRNA. The AUG codon signals both the interaction of ribosomes with the mRNA and also the tRNA with the anticodons. The tRNA, which initiates the protein synthesis. The next step is for a second tRNA to approach the mRNA. This is the code for proline, the anticodon of the proline tRNA, which reads this is GGC. The final process is to start growing peptide chains by having amine of proline to bond to the carboxyl acid group of methionine in order to elongate the peptide.
Step 3. Elongation. Elongation of the peptide begins as various tRNAs read the next codon. When the correct match with the anticodons of the tRNA has been found, the tyrosine forms a peptide bond with the growing peptide chain. The proline is now hydrolyzed from the tRNA. The proline tRNA now moves away from the ribosome and back to the cytoplasm to reattach to another proline amino acid. Step 4. Elongation and termination. When the stop signal on mRNA is reached, the protein synthesis is terminated. The last amino acid is hydrolyzed to form its tRNA. The polypeptide chain leaves the ribosome. The N-formal methionine that was used to initiate the protein synthesis is also hydrolyzed to form the completed peptide at this time. The ribosome is now ready to repeat this synthesis several more times.
DNA. Polymerase. What is that? DNA. Polymerase. What is that? What is that? I'll tell you. An enzyme that joins individual nucleotides to produce a new strand of DNA. DNA.
I will be explaining how to use this codon chart. You use a codon chart to translate your DNA sequence into an mRNA sequence, and you do this by changing the original strand with the appropriate base pair. And then you translate them from the original DNA sequence to the mRNA sequence. Once you have separated the mRNA sequence into groups of 3, you will look on the chart, and for example, let's say your first codon is CAU. So you'll start from the beginning and move out. You'll start at C and go to A and then to U. And your amino acid will be histidine. That will be your amino acid. And then this is another codon chart you can use to figure out your amino acids. So let's say you have AUG. You'll start where it says first base and find A, and then you'll go to second base, find U, and then you'll go over here and find G, and it'll be AG, and AG is a start, and it also is methionine.
There is one start codon and 3 stop codons. Transcription carries the mRNA across the DNA strand.
Now I will be doing a review on all of the key terms. First off, we have DNA polymerase. This is an enzyme that joins individual nucleotides together to produce a new strand of DNA. In the picture you will see next, it will show DNA polymerase. Our next word is RNA. RNA is a nucleic acid that consists of a long chain of nucleotides. In the picture you will see next, it will show RNA. Next we have messenger RNA. Messenger RNA is an RNA molecule that carries copies of instructions for the proteins. In the picture you will see next, it will show a picture of messenger RNA, also known as mRNA. Next we have ribosomal RNA. This is also known as rRNA. rRNA is a type of RNA that combines proteins to form ribosomes. In the picture you will see next, it will show a picture of rRNA. Next we have transfer RNA. This is the last of the RNAs. It's also known as tRNA. Transfer RNA carries out each amino acid to a ribosome during protein synthesis. In the picture you will see next, it will show a picture of transfer RNA in action. Next we have transcription. Transcription is a segment of DNA that serves as a template to produce RNA molecules. In the picture you will see next, it will show a picture of transcription in action. Next we have RNA polymerase. RNA polymerase is an enzyme that links together the chain of RNA nucleotides during transcription. This uses DNA as a template. In the picture you will see next, it will show RNA polymerase in action. Next are our introns. Introns are portions that are out or discarded from mRNA. In the picture you will see next, you will see a picture of introns. Our next word are exons. Exons are the remaining pieces that are spliced together to form the final mRNA. Our next word is a polypeptide. A polypeptide is a chain of amino acids. In the picture you will see next, it will be a picture of a polypeptide. Next we have a codon. A codon is a set of 3 adjacent nucleotides. For an example, we have AUG. A is 1 nucleotide, U is 1 nucleotide, and G is 1 nucleotide. But altogether, AUG makes 1 codon. AUG is also usually the start codon. Next we have an anticodon. An anticodon is a sequence of 3 nucleotides located on the end of transfer RNA, also known as tRNA. Lastly, we have translation. Translation is a step in protein synthesis when the genetic code is carried by mRNA and then is decoded to produce a specific sequence.